In the next section, we'll look at how you could detect in this very long, remember, 3 billion bases for a human genome. And there's species with much larger genomes. We're no exception. We're neither very short nor very long in terms of the genomic sequence. But 3 billion is quite a lot. How do you find out where there are genes or how what they do? And so I want to zoom in a little bit on this specific task. Suppose for some reason you've already managed to get the full genome. Like in our case, we have this since 20 years. It was co-developed, but suppose you just have the sequence of the whole genome and you don't know what to do with it. How could you try to identify these protein coding regions? That's by and large what a gene is for us. Is a region where this genetic code gives you a protein. And there's a few things, as almost always in bioinformatics, there's two options, which one is you just take this data as I mentioned, and you try to do something that's intrinsic just to that piece of data, that piece of information. I'll discuss the options in a second. The other option is you somehow involve other knowledge about the biology and collect additional information, maybe additional experiments in the lab and so on. Almost all real results in research are a combination of these, but it sometimes helps to keep things apart. We'll focus on this ab initio gene detection, where we're just taking the letters and try to figure out what we can say from that. And the first one that we'll focus on is this start and stop codon meaning in the code. Maybe it's worth going briefly back. So you notice there's one start codon, which by and large, you need this in the DNA sequence to start the translation of a gene or this translation of a protein. And then there's one of these terminating the translation. A sequence in the DNA that begins with one of with the single start codon and ends with one of these, and in particular doesn't have any other occurrences of the stop codons. That's called an open reading frame. And you should think of it as like this is kind of opening the book and this is closing it shut. So trying to read a gene immediately stops when one of these occurs, so you can't keep going. There's another thing that usually to start the translation, remember this fat brown blob that was kicking off the translation in the video, the RNA polymerase. This has to somehow attach to the DNA. And usually there's some, if you think about the DNA strand and say, here's the start codon, and then you keep reading and there's an end stop codon somewhere here. So usually there's a little upstream from the start codon, some specific sequence that allows this the RNA polymerase to attach and stick there. So you can also try to find these. Unfortunately, these are not always the same. So we'll come back to these in the latter part of the unit. They're the tricky bit is that they may be a different sequence in different species. There's a third one that's a little more subtle, and is one of the things that didn't make it into the module in the end. But there's, if you look at it statistically, there's a subtle difference in how often the different letters occur in a protein coding sequence and outside of these. It's usually too subtle to see with a naked eye, but you can train a specific stochastic or a statistical model to notice the difference. It's not an exact thing, so it's only a probabilities game, but you can use these. That's essentially hidden Markov models that you can use for that, if you've maybe heard the term, who knows. They can essentially give you a probability whether you're in the gene mode at this point or in the non-gene mode, and they can transition with some probabilities but it's a very fuzzy business. They'll only give you a hint. It's more likely to be here or more likely to be here. Okay. For today, we'll focus really on this first part. And you could ask yourself, is there any hope with just these start and stop codons? Is there any hope to detect genes in any somewhat reliable way? And what helps us here is that evolution is a certain directed force. Over long stretches of time, there's always going to be random mutations in the DNA. Just because these bases, I mean, we think of them as 4 distinct letters, but they're actually consisting of smaller molecules or smaller atoms. And you can convert from one to the other if you change some of the atoms, which sometimes happens by random chance. And also in the copying process, you can sometimes have mistakes, even though they're not so likely. So there's going to be these random changes in letters. And now 2 things can happen. Either this essentially destroys the functional part of whatever it was modifying. Maybe some gene now encodes for a different protein and this one doesn't function anymore. If that's bad enough, if that's fatal for the organism, this mutation will just die out. Right? This is no longer observable because all the creatures that had that mutation are no longer with us. On the other hand, there might be mutations that don't change things, and then these might still be visible. And there might be parts in the DNA where at least the exact function isn't clear, but for many parts we know there's heavy differences between different species and even individuals of the same species, and nothing really happens. So it's somewhat reasonable to assume that there's parts in the genome where mutations are important to avoid, and then these will be highly conserved. Whereas for the other parts, we don't have a reason to assume that, so probably they look somewhat random, because the mutations are dictated by random events. And if nothing in the evolution stops them from being selected against, then they will stay random. So we'll always compare what we see in some specific genome to a null model, which is that stretch of DNA is just generated randomly. And if we observe something where the probability to see it in the null model is very, very low, we can take that as a signal that we found something of importance, of biological significance, one way or another.